All right. So welcome everyone to our DUNES webinar series. And many of you may be returning from spring break, and I hope you had a good spring break. For today's webinar, we are very happy to have Dr. Ramesh Rao from UC San Diego. He'll be talking about health interventions based on decoding and modulation of electrical signaling in humans. And just a quick bio, Dr. Rao has been a faculty member at UC San Diego since
1984.
1984. My thing got cut off on my screen. Since 1984, he's the director of the Qualcomm Institute since 2001. He holds the Qualcomm Endowed Chair in Telecommunications and Information Technologies and is a distinguished professor in electrical and computer engineering. He is involved with a variety of research initiatives, leading major interdisciplinary and collaborative projects at various levels. And he has been on numerous health-related boards, including the UCSD Health System Advisory Board, the Academy of Neurosciences for Architecture, the Corporation for Education Network Initiatives in California. And he has a long history with, and he's been the director of Qualcomm. So without further ado, let me hand over to Dr. Rao so we can hear his wonderful talk.
Thank you very much. I appreciate the opportunity to share my personal experiments to some extent with meditation in particular. I'm trained as an electrical engineer, wireless networks, but in my role as the director of the Qualcomm Institute, I've been influenced by methods and practices and techniques from a variety of different disciplines, given the interdisciplinarity of the Institute. And that's kind of how I segued into trying to understand from a somewhat quantitative perspective, the signaling that takes place inside the human body as we go through different experiences, some passively and some actively through activities like yoga and meditation. So this is a little personal journey for me, but it's grown on me. And to some extent, I think there's a lot more that electrical engineers can bring to the study of topics such as meditation and maybe even consciousness than perhaps is currently the case. So that said, let me dive in. As somebody who studies network, one of the things that jumped at me as I was reading is that the human body has a built-in sensor network, which is always active. I believe it refreshes itself every 300 milliseconds or so. 80% of the fibers of this wandering vagus nerve basically conveys signals from the various sensors we have, receptors, chemoreceptors, thermal receptors, pain receptors, stretch receptors distributed all over our body. So it's for sensing, and the remaining 20% is basically to do something about it, the actuation of various processes that react to what we are involved with. Now, we can catch glimpses of the activity of that larger sensor network with heart rate monitors. So I want to emphasize that the autonomic nervous systems, parasympathetic and sympathetic branches, you've probably seen pictures of this sort, innervate essentially every visceral organ. Of these, the heart is the most electrically active. You can detect the heart's electrical activity rather easily. But the point of it is not just to study the heart, but to understand the whole body by using the signals that we can readily extract from the heart. So diving right in, the heart is a bit of a artist's rendition, is innervated by sympathetic nerves from the sympathetic nervous system, as well as the vagus nerve from the parasympathetic side. I'm sure you've heard this analogy. The sympathetic side is kind of like the accelerator, and the parasympathetic side is the brakes. The most dramatic way in which I have come to understand this is when a recipient of a heart transplant gets hooked up to a new donor heart. They don't actually connect up the vagus nerve. So there are no brakes, and this heart, this transplanted heart, beats very regularly at 120 beats per second because there is nothing that restrains it minute by minute, beat by beat. You see on the right-hand side, the classic PQRS, TU points of inflection of the electrical signals that you can extract with a ECG sensor. It's a superposition of the signals that radiate from the sinoatrial node, the SA node, as it propagates through the various chambers. The R peak corresponds to the left ventricular contraction. This is the action that squirts blood through the entire body. It's the strongest one. And it turns out that from a signal processing point of view, if you have access to this time series from one R interval to the next R, the peak, from one peak to the next peak, you can extract a lot of meaningful information by just analyzing that. One of my earliest sort of significant motivators was a study that basically looked at 24-hour time domain heart rate variability data. There's more than 24 different measures that have been used in the literature to study the variability of heart rate. And the first thing you learn is that a regular heart is actually a sick heart. A heartbeat which has a lot of variability in the RR intervals is actually a healthier heart. So depending on which measure you look, standard deviation of NN intervals, root mean squared of successive differences, or percentage of beats that vary from each other by more than 50 milliseconds, all of them go down as a subject ages. So a newborn has very high heart rate variability and an older person has very little. And in fact, there are clinically established thresholds that tell you when one is in a critical region. So if your heart rate variability slows down and you're a normal person, not with a transplanted heart, then that's cause for concern. So heart rate variability can be related to age. And I started to experiment with myself. You can buy fairly low-cost ECG monitors, chest straps, and other things like that. Early on, I was asking myself, what is it that moves me? And one of those is actually music of a certain kind. So what you see here, make long story short, is the same waveform, right? It's essentially the same waveform that was gathered. And what I was doing at this time was sitting quietly with my headphones on, listening to music. Music that comes from a certain tradition in India, kirtans. There's a spiritual dimension to what is being said. And what I was able to do was, in effect, look at the spectral components in the fluctuations, beat-to-beat fluctuations. And in this duration, during which one might think of as, a wandering mind state, you see all kinds of frequency components that are present. It falls off in a particular way, but nonetheless, they're all present. A little bit later, you begin to see things collapse around a certain frequency, okay? And then much later, it really got sharp. If you look at the y-axis also, the intensity of this is quite a bit more than what one saw before, right? I was taken aback. What was it about this music that I was listening to that resulted in this change in how my body was reacting to it? So let me attempt to jump into it a little bit. So this is a recitation. It's kind of like a spiritual chant in which at this point in time, there's a particular manner in which one attempts to define the divine, not by articulating what the divine is, but what the divine is not. So it's almost like a mental puzzle. It basically steps through a series of what it is not. It's like 25 different things that it isn't. And what is left at the end is what it is, right? So it really engages the mind. And this was quite startling that just listening to music could have such a strong effect. So off I went in search of other meditative experiences. This is a particular tradition. This was actually a rather large gathering at UCLA about 500 people, and we would sit together and we would chant various different chants, and sometimes there are hand movements or hand postures, sometimes there are breathing instructions. But this was a 61-minute long exercise where you sit across facing a partner. And here is the data. Really sharp peak. And if you go back to this diagram that I had shown you, and you map, the values of SDNN that I was able to measure on myself, RMSSD, PNN 50, the measured data for this 1 hour corresponds to an individual way younger than I was, a 30-year-old or a 10-year-old. So it would seem that whatever the heck we were doing, whatever this chant was doing to my body, it had a pretty profound effect in cranking up the vagal tone and altering my state of consciousness, which, by the way, is the subjective feeling when you go through this long meditation sessions. We would actually do this for about 8 hours. So this was just one of those 8 sessions. Now, this variability, the spectral components can be decomposed. There are time domain measures, there are frequency domain measures, there's all kinds of measures. It's a field in itself. But I was then interested, based on the previous experience, at trying to understand group effects. Are these strong phenomena taking place because it's not one person sitting alone and meditating, but doing it in the company of a large number of people? So I came upon this paper, which is not that heavily studied, which basically comes up with physiological meaning for spectral components in different bands. Let's see, there is a chat. Okay, very good. So what this does, low frequency on the left-hand side, of course, it breaks up the band into different regions. Respiratory sinus peak is because, when you breathe in, your heart rate accelerates a little bit. When you breathe out, it decelerates a little bit. So there is this synchronization with your breathing. The myogenic peak is because, the muscles of your, many muscles have this built-in effect that they will twitch by themselves, kind of like your heart muscles. But that's the frequency at which they do that. Neurogenic is the repetition rate of signals that come down your neurons. And then it turns out that your capillaries, there is a certain pulsation that takes place as it moves the blood around, and that too can be decomposed. So these bands, these colors that you see in the background correspond to activities, physiological activities of that sort. And the blue curve you see is actually my own data from a session that I wanted to see. Do I see these different peaks? And sure enough, you do. Which then, okay, got me going down this path without going into all the details. These are different kriyas, different meditative practices. And if you look at this figure, you realize that different meditation techniques have sometimes profoundly different effects on you, right? If you see the number of peaks, the magnitude of the peak, and the location of the peak, it seems like it's almost, you can think of it as your meditation medicine cabinet. You can kind of decide what form of meditation you might need, given the changes that you're seeking to generate. So it wasn't just this one uniform thing. So we actually collected some data here. The gentleman on the left, David Chanoff-Khalsa, a well-published researcher in a particular form of yoga, Kundalini yoga. And you see on the right-hand side a number of us. None of us, other than me, are from India. So meditation is something that's acquired based on some relatively brief period of training and practice. We wrote a paper about it, which has more of the details. But what we were studying is there entrainment? Do we couple with each other? And if so, in what manner does this coupling take place, the intensity of the coupling? So there are methods that I think, it's best if you were to look it up in the reference that I mentioned. But suffice it to say that what you see up here, A1 through A7, are different kriyas that we were doing. Time on the x-axis here. And in this periodogram, red intensity shows greater entrainment, blue lack of entrainment. So you begin to see, so this is pairwise entrainment between two subjects. You begin to see that there are certain points in time when one gets entrained and other points in time you don't. So we came up with a way to define this for the group, and now it sharpens. You can begin to see that there are these intense entanglements that take place during specific kriyas and not during others. There's another way of representing this where
Ramesh,
yeah.
maybe a quick pointer. There was one question on the chat, in the Q&A, if you'd like to answer now or hold off.
Let me see here. Let me bring up the chat. Probably have to stop share. Yeah, so hard math approaches. So synchronization, especially with RSA, yes, it's something that we've been playing around with using pulse ox sensors, but we have gone beyond what hard math's original approach was. Maybe at the end I can come back and address this question.
I think maybe in future I can just read out the question to make it easier.
Yep. All right. Do you see my full screen?
Yes. Yes.
Now I have to get my control back. Yeah. Okay. So what you see here are 5 subjects that were sitting together and meditating together, going through different kriyas. And what you see dynamically is the kind of entrainment. The color of the line, the thickness of the line is indicative of which band and how much power there was in that entanglement. So what you see is that there are kriyas in which you do get entangled quite significantly. This one in particular was quite interesting because you're actually not chanting. You are sitting quietly in that posture. If you see, you're kind of like squeezing your thumb into the top of your nose bridge. And interestingly enough, this is what the data showed. The respiratory sinus arrhythmia is what one might expect to be taking place because of synchronization of breathing. If you're chanting, maybe you're breathing in and breathing out in unison, and that might be one reason why you have that. But what you see in this chart is this coupling that takes place in all the other bands, the myogenic, the endothelial pulsation at a lower frequency. So there is some evidence, I think, even though it's a small study, that suggests that there are strong group effects that help with this. Okay, and now I'm going to switch to something different.
Maybe one quick question on my end, Ramesh.
Yeah.
So was there any relationship to how close these meditators were sitting? Was that affecting these couplings?
So for the data that you see here, we weren't sitting particularly close. There are other data sets we have where, like in that other picture, like in this picture, you're actually physically sitting very close, like this one. You don't touch each other, but you're like an inch away from each other, your knees. So yeah, you wonder what causes this coupling. Okay. So now I'm going to switch to something that we were briefly chatting about. As I mentioned to Sai, San Diego has a very large veteran community, many of whom are in their late 20s and 30s, and they come back from deployments with PTSD, post-traumatic stress disorder. And, this is a big field. There's a lot of literature here. There's also programs and techniques that are being employed to help people overcome PTSD. So a very top level sort of summary is, because of whatever the previous association is, there is a dysfunctional triggering of the flight or fight response. A smell, a sight, a sound is associated in the mind with a horrible incident, and it results in the flight or fight response. But if you had a chance to look into this polyvagal theory, it is a theory. The polyvagal theory basically says, as we evolved, this is my summarization, as we evolved as human biologically, we never let go of biological mechanisms from the past. So one of a third response to this triggering is actually playing dead. This is sometimes described as the dorsal vagal shutdown. A snail, for example, exhibits this. You poke a snail, it retreats into its shell. It doesn't try to get away from you. So this theory would argue that human beings also have this polyvagal shutdown, dorsal vagal shutdown mechanism. And we came upon an interesting subject with the Veterans Administration. This subject was a chaplain in the Marine Corps. So you might know this, but chaplains are there in the battlefield. And if a soldier is hit and about to die, they are there to minister to them. They hold them in their hands, so to say, so they're very close to them physically. They're not allowed to shoot. They do not carry weapons, so no fight. And they do not abandon their dying comrades, right? So there is no flight either. So an individual who spends their lifetime ministering to others who are dying and is trained not to fight or flight, what is left for them? Well, for this particular subject, they would pass out. So when their PTSD was triggered, they would pass out. They would throw up and pass out. So we were looking to see, so what happens is their daily lives are very badly affected. They cannot drive because you don't know what will trigger this. And it's kind of it's not a good life that they end up living. And even a few minutes notice. So if you can find out 5 minutes in advance that this is going to take place, you might be able to pull off the road if you're driving and, otherwise be able to drive. So we look to see if there are HRV-based predictors that might help do this. And it turns out that there is this particular measure, the LF, low frequency to high frequency ratio, that basically allows you to make this distinction, the basal condition versus after gagging. So there is a significant shift that takes place after this. So what we did was we gathered data from this gentleman, PNN 50, you can state it in terms of that number. You can also state it in terms of the normative age correspond to that value. And we had this gentleman keep a diary. So it says here, 6:15, wake up, coffee, 7:45, extreme coughing, gag, fall down, and recover, right? So he, of course, had no idea what the data was showing. But if you look at this 7:45, when he fell down, his PNN 50 was the highest. He's in his 40s or 50s. So actually, that moment where they gagged and fell down was a relief, okay, from the state that they were in. So this gagging reflex is partly there to restore a sense of balance. And it's a pretty horrible life. This gentleman is in his 40s, but most of the time, his physiological signals correspond to that of an 80-year-old. But here is the data that we were able to get, right? 5 minutes before, if you were tracking this in real time, look at the change, 5.71, and it goes down to 0.28, okay? Kind of like what the study showed, 1.7 and 0.6, suggesting that this mental state can be inferred by doing this real-time inference. I want to talk a little bit about what happens when you try to gather this data in real time. And the big worry that you have is errors, because these devices, these sensors that you have attached onto yourself might be moving around. And as a result, the data may be in error, your deduced measures may be in error. So that's the real point of what I'm trying to get at. But it's a little segue. Since this was wearable, one of my students, actually ex-student, was pregnant, and I was sharing this with her, and she said, "You know what? Why don't I collect some data for you?" She's pretty advanced stage of pregnancy. She was expecting to have her baby. So it's an interesting thing, the way the uterus works. There is this switch that gets thrown. This is blood markers, corticotropin releasing hormone binding protein, which prevents the uterus from going into contractions prematurely. But then at some point in time, it dips. And when these two numbers cross, that's typically when a woman goes into labor and child is born. And so it turns out that this LF over HF ratio is a way to track this CRH level. Okay, so it can help you predict pregnancy and delivery. And in some countries, including India, deaths in young women during labor is one of the highest causes of mortality. So it could be very useful predictive tool. So anyway, this student of mine wore this for a long time, and this is what the data looked like. Looks like a mess. Looks like there was a lot of error. So I was disappointed, and I said, yeah, it doesn't look like there's anything useful. She said, let me look at the data. And she looked at the data, and then she had a pretty startling observation. She said, what looks like noisy data here and there, this is when the fetus was moving. Okay. But then it hits you that basically there are two hearts beating. And what you're observing, what you're measuring with one sensor is a superposition of these two waveforms. And to some extent, because they intersect each other, your RR intervals are not accurate. Here's another example. This is data that I gathered on myself when I go running in the mornings. And I would notice that sometimes in the first few minutes, there would be this very high heart rate, like 196, and then it would drop down to 138. And I kept wondering, what the heck was this? Why do I see this increase? It wasn't always there, but there were a number of times when it was present. So I then decided to put on an ECG monitor, and there's an interesting thing that happens. I hope you can see this waveform here. There is this R waveform, and there is a T peak. So it turns out that many of these devices, I think, use a level crossing detector. I don't know if you can see this purple line here, okay? There are these really skinny green inflections that correspond to the R peaks. This is actually a T peak, where I'm hovering. This is a T peak. This is a T peak. But the T peak sometimes goes above the R peak. So instead of seeing two R peaks, it sees 4, right? So it seems like it is catching something in error. So what do? Discard this data, or is there some insight behind this? Well, it turns out that in the early few minutes when you start to run, there is a lot more potassium that gets released in your blood because of the deployment of glycogen from your muscles. So the level of potassium in your blood goes up. There's a study that documented this by collecting data minute by minute when runners were running. And it is also known that when there is excess potassium, then it leads to T wave elevation, okay? So in some ways, what looks like an error is actually revealing something else about what's going on. Somewhat excessive release of potassium. And if I go back, so I also try to manipulate my diet. If I go back, what I did between here and here, this is March, this is August, was basically manipulate the potassium level, a little bit more sodium, a little bit less potassium. So sometimes what looks like errors might actually be revealing something deeper. And so if you want to use methods that simply discard errors, you might be throwing out some interesting stuff. So that was a summary, glimpses of stuff based on HRV. But we also wanted to understand how other forms of stimulation, electrical stimulation, both sensing and stimulation, would affect. So here is a pretty cheap device that you can use to stimulate your vagus nerve. This is one of the places where it's easily accessible. And it alters your physiology. But rather than go over this, let me share an accidental recording that we once made. I started recording around 5 o'clock in the evening, and I stopped recording at about 10 p.m. This is the duration of time when I was electrically stimulating my vagus nerve. Okay, that's Dr. Lerman. So it was interesting you said that you're also working with people in anesthesiology. He's an anesthesiologist, among other things. And you see this electrode on the scalp, which is confirming that I am one of those subjects for whom the vagus nerve stimulation, electrical stimulation does register. Apparently, there are some people in which it doesn't have an effect, and you can tell that. So what do you see here? You see some activity while there was stimulation, but after that, the more squiggly the lines, the greater the heart rate variability. So kind of, I was able to induce this state of, a higher vagal tone through this electrical stimulation. And then life went on. And that evening, at this time, I went for a yoga class. Now, if you even eyeball what you see in this left blob with what you see in this right blob, they kind of had the same effect. So it seems like what this produces with electrical stimulation can also be produced through meditation, right? Measurably. Another interesting data set that I came across by trying it out on myself was, during a workshop at UCSD on compassion meditation. And I did not know about this literature while I was participating in it. I just collected data. This was the RR intervals, whose absolute value was trending up during the lecture. And then during this period of time, when the RR intervals are trending down, we were actually practicing compassion meditation. The way it worked is you turned to the partner next to you, you looked into their eyes, and you were imagining in your mind a concrete form of compassion. And what I was doing literally in my mind was recalling this image, which for some reason sticks in my mind. It's just an image of an abandoned child in Nanking, during the Japanese invasion. And, in compassion meditation, compassion, as you might know, if you're in the Buddhist tradition, is not to be confused with pity. It is actually a call to action, to move yourself to do something about it. So if you put yourself in the right mental state, then later on I discovered that actually people who have done this analysis, this is their study, is that your heart rate actually goes up during compassion versus neutral states. And so this is literally what happened, okay? But it's an interesting thing that there is a bounce back effect. So it ended at this point in time, but I continued to record. And at this point, it bounces back and the heart rate reached a low of 37, which was quite amazing. So this mental exercise, no breathing, no other cues, no hand movements, had this pretty profound effect. So you're further drawn into the mind. I want to acknowledge there is a large team of people who are involved in the results, the data that I'm going to discuss. Dr. Lerman is our fearless leader, anesthesiologist, formally speaking. But along came the pandemic, and we got this award from BARDA. And what this effort does is basically recognize that many people that were dying in the early years, early months after the pandemic, were dying because of a cytokine storm that takes place. And you have all these fluids that accumulate in the lungs because of this dramatic increase in the release of cytokines. And was there a way in which the vagus nerve could detect the release of these cytokines, right? So you wouldn't need, in principle, to do a blood test. You could sense using electrical and magnetic sensors. And that was what this project was about. So if you don't mind me jumping ahead, so it turns out that depending on the class of pathogen that an individual is exposed to, by the way, so the most important thing that I think you need to notice here is that the vagus nerve fires within seconds to minutes, right? So you don't have to wait for a bunch of time to elapse. You certainly wouldn't need to take a blood test and have it done. So it is sensitive enough. So this is blood work. The x-axis is blood markers. Think of it as blood tumor necrosis factor, interleukin-6 that are expressed. And you can see that there is a distinction between the levels of these things that are expressed as a function of the class of pathogen. And then also the degree of exposure. So you can you can see what this looks like. But the part that is of greatest interest is basically, are you able to detect this through signals that you might be able to detect off of neurons that run through your limbs in particular, right? And it turns out that the answer to that is yes. And that's what this effort is about. But if you want to make these measurements in free living conditions, it's a little bit difficult because these signals are very faint and there is noise in the outside world where you're trying to make these measurements. So coincidentally, we have been fortunate to have rebuilt MEG facility. So this MEG chamber is not very big, but not very small. It's about 9 feet by 13 feet, about 12 feet. It has 2 feet of magnetic shielding all around, right? So it cuts out the Earth's magnetic field. So you can see this chamber under construction. This is a really heavy door that you install, and it gets sucked in kind of like a bank vault. But at the end, it produced a chamber which the Earth's magnetic field in La Jolla is approximately 45,000 nanoteslas. And down inside this chamber, in the middle of this chamber, it's down to approximately 1.7 nanoteslas, right? So you cut out the magnetic signals, the noise from the outside, which then allows you to use an MEG machine. There's only 2 of these in California, apparently only 25 of them in the country, that passively detects. There's 300 sensors. This is a cryogenically cooled chamber with squid sensors that passively detect magnetic signals coming off of the brain. And you basically do the source localization. Its resolution is about 1.5 millimeter cube into the brain. Temporal resolution is 1 kilohertz. So it gives you a lot of spatiotemporal details. And inside this chamber, there are mechanisms. There's a screen, so you can subject the subject to visual stimuli, auditory stimuli, tactile stimuli, haptic stimuli, and watch how the brain reacts. So completely passive, no radiation, no exposure of any kind. If anything, everything is shut down. But it does require you, this is to cost us about $1.5 million to set the chamber up, and another $2 million for the MEG device itself. And it's a pretty complex approach to sensing what's going on. But along came this other innovation, optically pumped magnetometers. At the heart of an optically pumped magnetometer is this small little vapor cell, in this case, rubidium atoms, and you pump it using a circularly polarized laser beam, and it aligns the direction in which these atoms point. So that's the pump beam. You then use a probe beam and you sense the probe beam, and the deflection of the probe beam depends on which direction in which these atoms are aligned. So if you have an external magnetic field, which you see here, dotted lines, then they kind of follow that, and you can detect the magnitude of this external signal by watching this. So what is interesting about this is that each sensor, I'm wearing them, there's like 7 of them on me, they're that small, approximately a centimeter by centimeter by centimeter, okay? and you can attach them all over. So here's what it looks like. Now, this data was taken inside the chamber. My head is not inside the actual device with the squids. So it's just the magnetic shielding that is being exploited. That's what it looks like. And what I did was gather some data on myself a day before I took the COVID vaccine and the day after, right? So the COVID vaccine is kind of like the inflammatory stimulus. And then I'm in a position to understand what various activities do, right? So in the beginning, I would sit there in a resting state. So the direction is just relax, no further instructions. Then you go into a paced breathing, okay? And then you get into a meditative state. They kind of lasted about, the meditation itself is half an hour. The others together lasted about a half an hour. Now, there was a very specific form of meditation that I was practicing, about which there is a study. There's a published study. And what they did in this study was individuals who practiced this for half an hour every day had blood work done to see pro and anti-inflammatory expression of these various cytokines before and after the study. And what they found is that this particular kriya actually reduces the pro-inflammatory ones and increases the anti-inflammatory ones. So there's some data. And of the many kriyas that one could use, I said, why not try this? So here's how it actually goes. You're chanting, okay, and you're tapping your fingers. That's basically what you're doing. For the first 5 minutes, you chant loudly. The next 5 minutes, you chant in a whisper. The third 5 minutes, you do not chant audibly, but you're chanting in your mind, and then you reverse your way out of that, right? That's what you're doing. All right. So here is the data on heart rate variability pre and post vaccination. So yellow here in the HF band, indicates high vagal tone. So this is day before the vaccination, pretty good vagal tone. After the vaccination, the vagal tone is kind of gone. You're in a state of arousal because of the vaccine. So this is resting state data. This is a quantification of the resting state. If you look at these numbers, really significant drop from pre-vaccination to post-vaccination. And I have a number of these measures, SDNN, RMSSD, PNN50. These are standard things that are used in the literature. So there is no doubt what the body is going through. Now, this one gets a little bit more interesting. This is pre-vaccination resting state versus meditation, right? No vaccine in the picture. So up here is resting state, pretty high vagal tone. When I did the meditation, it actually reduced the vagal tone. So on this occasion, when there was an abundance of vagal tone, this meditation reduced it. Post-vaccination, if you look at the resting state, it was low in the resting state, but the meditation state actually increased. You see more yellow here than there. So in the previous instance, it decreased, the meditation decreased it. In this instance, the meditation increased it. It's sort of attempting to balance you, which is what it's supposed to do. This one summarizes more in a tabular form the effect of the meditation, right? So you see the higher the SDNN, the greater the vagal tone. Pre-vaccine, meditation reduced it. Post-vaccine, it increased it, right? From 70 to down to 43, then it went up from 20 to 34, right? So here is this kriya, which seems to be effective, at least in my instance, at leveling things. So we also started making some, we were also gathering data using the OPMs and trying to get a deeper understanding on, the search here is to be able to eavesdrop on the signals and figure out what signals are traversing these nerves that might give you some insights on how exactly does the body signal the regulation, up or down. I don't want to go into too many of the details here. So let me just shift here. So these are the actual waveforms. The blue ones are on day 1, and the red ones are from day 2, day 1 before the vaccine, day 2 after the vaccine. They're kind of similar. So there's lots of signals that flow down the nerves. But you notice that if you pretend these two are essentially the same, day 1 resting to day 2 resting, there is this enormous increase. So this particular waveform seems to code information about the inflammatory state. At the same time, this one actually decreased. So you see a lot more of this and you see a lot less of this. Meditation, you find these other signals that display an increase in one instance and a decrease in the other instance. So it was kind of like a search for what waveforms seem to be bearing information of what kind as they flow on the nerves. Okay, so looking forward to any questions that you might have, but the journey for me personally has been to kind of evolve from the capabilities of the sensors to something a little bit more abstract. There is a study that Laura Schmalzel, who used to be here with us, published, which basically tracks how different yoga practices activate, network, or attenuate the connections between different regions of the brain. So it seems like there's lots of possibilities, many permutations and combinations of how different parts of the brain talk to each other. And through these practices, you can alter it. And now with these sensors, you can sense whether those alterations are actually manifesting themselves in an objectively measurable way. I think progress here is limited by how many experiments you can do on yourself or in small groups, and the kind of analysis techniques that you need, especially when you're dealing with wearable sensors that are somewhat error prone.